Good evening, everyone. Please make sure that you let me know in the chat section whether the audio and the video is clear to you or not. Welcome to another session. So, from today till our main examination, we'll be revising and we'll be practicing out the previous year questions. So, if any one of you who is lagging in the practicing of the PYQs, this particular session on daily basis will help you out. Please confirm in the chat section whether the audio and the video is clear to you or not. We'll be revising, we'll be discussing, and we'll be practicing out the questions for the same. Okay, let's begin with the class now. Before starting, if you're new to our application, if you're new to our channel, make sure that you hit the subscribe button. In addition, make sure that you download our application, which is available both on the Android as well as in the iOS. So, make sure that you download it. We have a full course ongoing for our June 2024 examination as well. Any one of you who's interested or who is preparing for this particular examination for the June 2024 session, you can download our application and get yourself enrolled. It's an all-in-one course with all the required information, with all the required content available to you. So if you wish, you can enroll in the same, available in very affordable prices, right? All the informations are in front of your screen. In addition, you can get it from the application as well. In addition, we have a mock test series going on for the December 2023 examination also. So if you wish to find out, or if you wish to solve out the PYQ, solve out the mock test before your main exam, these will help you out. Let's begin. So, this is the very first question. The question is, iron sulfur clusters are the key prosthetic groups that carry the electron in all of them below, except which? First one is NADP coenzyme Q reductase. Then you have succinate coenzyme Q reductase. Third is cytochrome C oxidase. And third one is coenzyme COQH2 cytochrome C reductase. Question is asking you which of the following carry the electron, which of them have iron sulfur clusters and carrying electron. All of them have it except for one. What will be the correct answer for this one? Simple question. This is from your respiration concept, right? That we study in unit number 6 and 1. These are, and the respiration is an overlapping topic for both units, right? So if you look into the first option is, like, if you go for this question in odd one out scenario also, three of them are reductase, one is an oxidase, so you can eliminate this option number C, which is the correct answer for this particular question. Very simple question. NADH coenzyme Q reductase, COQ reductase, second is also reductase, fourth is also reductase, third one is an oxidase, which is the wrong option, that does not carry electron. Remember about this. C is the correct answer for this particular thing. Next question, which of the following gases is present in the stratosphere at a concentration which is higher than its concentration in the troposphere? This is a basic general science question. Which of the following gases is present in the stratosphere at a concentration which is higher than its concentration in the troposphere? First option is nitrogen, second is oxygen O2, third is ozone O3, then you have carbon dioxide. Question is, which of them is present in the stratosphere at a concentration higher than its concentration in the troposphere? Simple question, basic question. C is the correct answer for this one. Ozone is present in the stratosphere at a concentration higher than the concentration in the troposphere. So remember about this as well. Very easy question. All the questions that we are doing today are previous years itself. Okay. One more question. Again, this is a repeated question that gets uh, one or two times this particular thing has been repeated. Question is, historical frequencies of fires in an area can be determined by which of the following? Question is, how can you determine that at what frequency the fires in our area is being, we can determine the frequency of that, right? First option is radioactive dating of the tree remains. Then you have examining the fire scars and the growth ring of living trees. Then you have measuring the carbon content on the soil surface after the fire. Fourth is examining the records of the evacuation history of the nearby villages, right? Simple question again. Give it a try to this. Then we'll solve it out together. Yes, try the question, everyone. Mark the answer in the chat section. Give it a try at least. Simple question, basic question. Very good, Selva. Others also try it. Mark the answer for the question. Very good. Others also try it out. Question is, which of the following can be used to determine the frequencies of a specific area's fire, right? First is, like, you can eliminate first and fourth option very, very easily, not required. Then second option is saying examining the fire scars, and third option is saying measuring the carbon content. See, you can measure, or you can have the historical frequencies of the same with the help of both B and C, that is examining the fire scars in the growth ring also, and also via measuring the carbon content. But out of the two options that I have, if I want to mark the answer in a much more appropriate manner, my answer would be option number C, because it's a much more logical and a much more better appropriate way for measuring the carbon content, right? So remember about this. Measuring the carbon content in the soil after the fire is a much more appropriate answer. In addition, if you get any other question in the examination where all the options are there, but if C is missing, then you can mark the answer as option number B as well. But if both measuring of the fire scars and measuring the carbon content will be available, you'll have to mark the answer as option number C, right? Remember about this particular thing. Easy, important, and can be repeated in the upcoming examination. This one is June 2021, Selva. June 2021. All the questions that I have taken up, they are from June 2021 morning shift paper. Okay. I hope it's clear. Very easy question. One more question. Consider the four geological periods. It is asked which of the following, like, you need to find out the correct representation or the correct arrangement of the complete geological period from the earliest to the recent one. You have Quaternary, Cretaceous, Jurassic, Cambrian. Try it out, everyone. Very easy question. This is from your evolution concept, last topic of evolution, that of evolution topic that we study is the geological time scale itself. And every year you can expect one question from here. Okay, every single year we have a question asked in somewhere or the other from the geological time scale. In 2023 also we got a question, in 22 as well, 21 as well, and also in 2020. So from last 4 years consistently questions are getting asked from here. Mark the answer for this. Try it, everyone, at least. Very easy. Very good, everyone. Try it out. Very easy question. Quaternary, Cretaceous, Cambrian, and Jurassic. Simple question. Okay, let's see. So, what should be the correct answer? The correct answer should be D, C, B, and A. In the reverse manner, the answer is correct. First is Cambrian, then Jurassic, Cretaceous, and Quaternary. Make sure that if you're good in cramming, if you're good in remembering the facts, try not skipping the geological time scale. Okay, because you can easily score 2 marks and sometimes 4 marks as well. This one was a part B question, right? In addition to that, It can be asked in some other format in part C as well. So do not skip this particular topic. Next question, let's see. Question is progression from G1 to S boundary. When we learn about the cell cycle, you have various transitions learned, right? So progression from G1 to S boundary followed by the entry into the S phase. It's promoted by the activation of which of the following protein complexes. First one is CDK4 cyclin D, then you have CDK2 cyclin E, then you have CDK4 6 and cyclin. Fourth is CDK4 6 and cyclin D and E both. Yes, Kirti, we have negative marking as well. You have part A, part B, part C. In part A, 20 questions in total, 15 need to be solved. Every question, 2 marker, negative 0.5. Part B, same, 50 questions. Over here, you have to solve 35, 2 questions, 2 marks per question, zero point 0.5 negative. Part C, 75 questions, 25 need to be done. Every question 4 marker, 1 mark negative for every wrong answer. Okay, this is the marking scheme for the entire paper. A, B, C, 3 parts are there. A is general aptitude, B and C are theory segment. No matter you are from botany, zoology, life sciences, you have to give the paper in life sciences stream only. There are 13 units in total. Everyone mark the answer for the question. G1 to S boundary followed by the entry into the S phase. So it is asking us about the activation of which of the following protein complexes is will be done. Very good, Selva. Others also mark the answer quickly. CDK4 cyclin D. Then you have CDK2 cyclin E, CDK4 6 and cyclin, and then you have CDK4 6 cyclin D and E both. Mark the answer. Very good, very good. Everyone mark the answer for the question. CDK stands for what? CDK stands for cyclin dependent kinases. B is the correct answer. CDK2 and cyclin E. It is asking about. Now, Selva, see over here. It's very, very easy for us to mark the answer as CDK4 and cyclin DE. Reason being, when we learn about the progressions in specifically the G1 to S phase, what do we see? We see the transition going on with the help of two different formats, right? First, there will be partial phosphorylation, and then we have the complete phosphorylation, right? In partial phosphorylation, you have 4, 6 and D helping in, and in the complete phosphorylation you have CDK2 and cyclin E working in. So it's asking us about the progression from G1 to S boundary, and then entry into the same, entry into the other one will be promoted by activation of what? So promotion by promoted by the activation of, like, when the complete phosphorylation will be getting done. So it is CDK2 and cyclin E. Okay, because in this scenario they are not talking about the important segment which is your CDK2, right? They're not talking about CDK2 over here, which should be there. So CDK2 and cyclin E will be a much more appropriate segment to mark the answer, right? Because 4, 6 and D, yes, it's there, but if by chance our CDK2 does not get activated, there are chances that the promotion into the S phase will not be done. So B will be the correct answer for this question, right? So remember about this. Second is the correct answer for this. I hope it's clear. One more question. Let's see. Very good, Anjali. Correct answer. Next question. Following statements are made about uncompetitive inhibition of the enzyme. When we learn about enzyme kinetics, you have three major types. You have non-competitive, competitive, and uncompetitive. In each of the three scenarios, there's a change in Km, Vmax, and Km in a specified manner. Same question is asked over here. First option is in the uncompetitive, the inhibitor is binding to both the free enzyme as well as the enzyme substrate complex. Second option is saying addition of uncompetitive lowers the Vmax. Third is saying the apparent Km is lowered. Fourth is saying the apparent Km remains unchanged. You need to find out the combination of the correct statement. Very easy question. Simple, straightforward, basic question. You just need to make sure that you know about what or how does the inhibition occurs in case of your uncompetitive inhibition, and how does the Km and the Vmax changes. If you have been enrolled in our courses, you won't find a difficulty in solving out such a question, because we focus upon the main pointers from where the questions are asked, and solving out the questions then become easier in the examination. Try the question, everyone. Very good, Anjali. Very good, Roshni. Others also mark the answer. Okay, let's see what will be the correct answer. First option is saying uncompetitive inhibitor binds to both the free enzyme as well as the enzyme substrate complex. No, it binds only to the enzyme substrate complex and not to the free enzyme. This is the main important property of your uncompetitive inhibition. Remember, in uncompetitive, what do we have? In uncompetitive, your inhibitor is getting bound to your enzyme substrate complex. In case of your non competitive, it binds to the allosteric site. That is any other site on the enzyme structure except for your active site. That is what an allosteric site is. And in case of your competitive, it competes with your main substrate to bind to your active site only, right? So remember about this. Competitive to the active site, non-competitive to allosteric, and uncompetitive to the enzyme substrate complex only. If I eliminate the first option, I can eliminate three options and I can reach to my correct answer without even solving out or without even reading any other thing to my correct answer, and that is option number A, B, and C. Then you have addition of uncompetitive inhibitor lowers the Vmax. This is correct. Vmax gets lowered and you have the Km getting lowered. It does not remain unchanged. It changes. It becomes lower than the normal scenario. Make sure that you recall about that how does the Km and the Vmax changes for non-competitive and the competitive as well, right? So one is the correct answer for the same. Remember about this easy, important, and scoring part. You might get a question repeated in the upcoming examination also. Remember, one is the correct answer. Let's discuss this particular question. The graph below shows the change in the size of the population of four different populations. Okay. A, B, C, and D. You need to find out which among the four populations would have the lowest intrinsic growth rate. You need to find out which will be having the lowest intrinsic growth rate among the four populations. Now, over here, you need to recall what is R, what is the formula for R, and how you can apply the same in this particular question. Try out the question, everyone. Very simple again. Mark the answer, everyone. Very simple question. What do you mean by R? R is your intrinsic rate of increase. What is the formula? Formula for the same is B minus D. That is birth rate minus death rate. Question is asking you which of the following would be having the lowest birth rate minus death rate. Which of the following will be having the lowest? B minus D over here. So this is birth rate, this is your death rate. In the first case, the population size is increasing with time. Increasing with time means birth rate is more, death rate is less, so it will be having the maximum R. Second one will be having birth rate again more, death rate less, because we can see a increase. Third is having equal birth rate and death rate, so no change. Fourth will be having, or the population is decreasing with time, so birth rate will be lesser, death rate will be more, so it will be the least over here, right? Because this is what you have to find out. You need to find out the lowest intrinsic rate of the population growth. So in the fourth scenario, in this particular scenario, what do you have? You have the, because when do we see a population size declining? See, on the y axis you have the population size, on the x axis you have the time. So with the passage of time, the population is declining, right? What is happening? The population is declining over there. So if it is declining at that particular time, that means my birth rate is lesser than my death rate, right? So in the fourth scenario, in the D scenario, you'll be having the lowest or the least intrinsic rate of the population growth. Is it clear to all? Yes or no? Just check. Very good, Anjali. Out of all the students, you were able to mark the answer correct. Excellent. Everyone check whether it's clear or not. You just have to imply the concept of intrinsic rate of increase or intrinsic rate of your population growth. There are two ways in which we showcase the population growth. One is your logistic, right? The second one is exponential. Exponential is based upon R. Logistic is based upon the carrying capacity, right? Carrying capacity is calculated via a different formula, and intrinsic rate of increase is via B minus D. We have learned all these things like in our enrolled batches. We have learned these things in detail. Everyone, is it clear to all? Just check. Very simple question, two marker. You don't require such a long calculation in the exam. You can just observe the graph then and there and mark the answer within a second. Very simple question. Right, let's move forward. D is the correct answer for this question. They could have asked the question which of the following is having no intrinsic rate. Like in which of them do you see a constant rate, right? Over here they have asked about the lowest. They could have also asked about the constant rate. So constant would have been option C. They could have asked about zero intrinsic rate. That could have also been the answer as C. They could have asked the answer as the highest intrinsic rate, right? They could have asked about the highest intrinsic rate also. Then A would have been the correct answer. Or they could have asked about the pattern, or they might have asked about the, what do you say, order in which the intrinsic rate is increasing or decreasing. So they would have like the question could be framed in any manner. You should know that how to analyze such questions. So analysis of the question is important, right? I hope it's clear. Let's move forward. Next question. Photochemically generated ATP is consumed in which of the following phases of Kelvin Benson cycle? Very easy again. It's asking you photochemically generated ATP is consumed in which of the following phases of Kelvin Benson cycle? That means it is asking you in which particular in the C3 cycle, at what step do you have the utilization of ATP? Mark the answer for this quickly. Very easy again. Very easy again. Question. Mark it, everyone. Okay, first option is only carboxylation. Second option is only regeneration. Third option is carboxylation and reduction. Fourth option is reduction and regeneration. Let's see the correct answer. Very simple. D is the correct answer. If you have learned about the C3 cycle, it's not difficult at all. So, in the reduction and regeneration step, we have three steps in our C3 cycle, right? You have carboxylation, you have reduction, and then you have the regeneration going on. So in the second and the third step, reduction and regeneration, we have the utilization of ATP. Very simple, not a difficult question. One more question, give it a try to this. Tumors are generally classified by which of the following? Tumors are generally classified by which of the following? First option, the virus which caused them. Second, the person who discovered them. Third, their metastatic ability. Fourth, their tissue or the cell of origin. Very, very simple question. Very good, Roshni. Everyone, mark the answer. Very basic question. You can easily score 2 marks out of this. And if you have observed the last 6 to 7 questions that we have done, every question is scoreable. Okay, every single question is scoreable. There's nothing which you should have, which you would have eliminated or like not studied in your preparation, whether with us in our application or yourself, that is self preparation. Very good. Very, very simple question. Tumors are classified obviously based upon their tissue or the cell of origin. All other three options are not related to the way in which we classify them. So remember, we have the tumors getting classified based upon the tissue or their cell of origin. Remember this. Okay, give it a try to this. Again, very simple question. Movement of the protein bigger than 50 kilodaltons require the nuclear envelope. Yes, the movement of the proteins bigger than 50 kilodalton across the nuclear envelope require which of the following? Sec 61, Tom 20, Tim 44, and then important B, beta. Very, very simple again. Try it out, everyone. Very basic question. If you have learned about the nuclear transportation, you will not find the question difficult. Mark it. Okay, let's see. Very simple again. So, movement of the protein bigger than 50 kilodalton across your nuclear envelope require important beta. Remember about this because in the nuclear transportation, beta important family of proteins are involved, which includes your importance and which includes your exportins. Both important and exportin, both of these are involved. Tim and Tom, these two are related to your mitochondria. right? They are related to the mitochondrial transportation, and sec 61 is related when we learn about the protein sorting segment. Like in your cell biology there's a concept of protein segment. At that time there's a concept of utilization of sec 61, so that is also not involved. Okay, so Tim and Tom in mitochondria. In the case of your nuclear transportation or the movement of the proteins across the nuclear envelope requires important theta. Is it clear to all? Yes or no? Just check. Very simple question. Translocon. Very good, Anjali. Very good. You are revising things properly, right? We have learned about this thing, right? In the protein sorting, when we learn about the specific protein reaching to the ER, how does the entire processing occurs, right? We have learned about this in our class, right? So B, C is the correct answer for this question. Very, very simple, very basic question, right? So just let me know whether these type of sessions are going to help you out or not for the till your main examination. If yes, we'll continue with the same sessions. Otherwise, we'll see into the revision of some other things, right? Yes, just let me know. Would you want me to continue these sessions till the main examination? And also, we have a full course running. If you're planning out for June 2024 paper, we have a full course running for on our application. All these things will be provided to you. Hard copy notes will be delivered to your doorstep. In addition to that, all the units, including general aptitude, will be available. All recordings, class PDFs will be available. Daily practice questions. We do questions in the class on daily basis. PYQ session, weekly test, regular doubt session, all these things in a single course. So it's an all-in-one course, right? And we'll be continuing with the same like question series every single day. 8 o'clock sharp. Be there on YouTube's live section, and we'll be solving out questions every single day. Hope this particular session was helpful, right? So be there in the class tomorrow also, and we'll be discussing the same. Okay, bye everyone, take care, all the best, and let's meet in the next class.